Hi everyone, I hope you are staying safe and tight. So I'm very glad today to record this talk, and thank you Cell and Developmental Biology virtual meeting platform for giving me this opportunity to present my work here. So today I'm going to present my work that would contribute to one of the fundamental biological questions, that how does organisms control organ size and what are the molecular bases of organ size regulation. So the topic of my today's talk is SID or JIP3 controls tissue size by regulating Diap1 protein turnover downstream of Yorkie gap. So how does organisms control organ size and the molecular basis of organ size regulation? So these are very puzzling questions, and these questions are the main theme of our studies. So broadly speaking, there are two types of processes that regulate organ growth. So systemic signals and the intrinsic factors. So systemic signals such as insulin and growth factors play a key role in determining body and organ size. For example, in Drosophila, starvation or genetic mutation in nutrient sensing gene produce animals with smaller organs. So these mechanisms are relatively well defined and are evolutionarily conserved compared to the intrinsic mechanism. So there are internal mechanisms that control organ size. So for example, a series of transplantation experiments in salamander and in Drosophila have revealed about these intrinsic size control mechanisms. So in contrast to systemic signals, so these intrinsic control mechanisms are not well understood. So genetic screens in Drosophila have identified mutations that deregulate growth in Drosophila's system, which led to the discovery of key and broadly conserved tissue intrinsic growth control signaling pathways, including the Hippo pathway or JNK pathway. In fly and mammalian tissues, Hippo signaling is conserved and potently controls tissue size by modulating the activity of genes involved in cell proliferation and cell survival. So JNK scaffolding proteins. So JNK signaling is mediated by JNK scaffolding proteins, which play a central role in cellular responses to apoptotic or proliferative signals. So Sunday driver SID or JNK interacting protein JIP3 belongs to that family of conserved scaffolding proteins that bind to and facilitate JNK signaling. So SID or JIP3 have been implicated in organ size regulation in mammals, but how does JIP3 regulate organ size? It remains unclear. So we took advantage of Drosophila wing, which is a powerful system, to address these growth-related questions. So to test this hypothesis, if SID regulate Drosophila organ growth, we knock down SID in developing wing epithelium using Drosophila gene-inducible system known as GAL4-US system. So when you knock down SID in the wing tissues, in the wing epithelium, so this resulted in smaller adult wings right here. If you compare these purple wings with these control wings, brown wings, so this is a significantly smaller wings compared to the control. So this piece of data, it suggests that SID regulates Drosophila wing size. So this is very interesting and exciting, as JIP3 loss of function mutations are associated with brain size defects in mice and human. But how does JIP3, it regulates tissue size? So we sought to determine the mechanism. So just giving you a background information about the Drosophila wing imaginal disc. So it consists of a posterior and an anterior compartment. So the patterning gene and glial is specifically expressed at this in the posterior compartment of the wing disc, where it induces the hedgehog expression. So hedgehog protein is trafficked to the anterior compartment, where it activates cubitus interruptus CI, which is the glee version of vertebrates. So in turn, this CI, it stimulates the expression of DTRAF, which is a component of JNK signaling in Drosophila. So this whole event, it leads to the JNK signaling activation at this AP boundary. So this localized JNK activity acts via the limb domain family protein ajuba to repress hippo signaling throughout the entire tissue. So it is unclear how this local effect on hippo signaling is transferred throughout the whole tissue to promote tissue growth. So we asked this very important question whether SID regulates wing size via this CI-JNK hippo signaling cascade. So to address this question, we specifically knocked down SID at the AP boundary cell where JNK is normally active. Here I'm showing in green. It reduced wing size. So if you compare the SID RNAi knockdown at the AP boundary, so compared to the control SID RNAi, it reduces the wing size. So this observation, it raised the possibility that SID regulates wing size via JNK. So to directly test this, we performed genetic epistasis experiments between SID and JNK. So we expressed a potent JNK inhibitor to block JNK alone or in the presence of SID RNAi in the AP boundary cell and determined the effect of these genetic manipulation on adult wing size. So as expected, vascular dominant negative, it reduced the wing size compared to the control. So importantly, blocking JNK failed to exacerbate the small wing phenotype caused by SID knockdown. So instead, these wings, it resembled the SID depleted wings. So moreover, when we overexpressed the wild type version of SID, it restored the vascular dominant active small wing phenotype. If you compare the quantification right here, so SID overexpression, it restored the SID RNAi phenotype right here. So collectively, these findings argue that SID is epistatic to the JNK and that SID, it functioned downstream of JNK. So as I explained earlier, that JNK acts via limb domain family protein ajuba to repress hippo signaling and promotes wing growth. Ajuba represses hippo by sequestering warts to cell-cell junctions. So warts is a negative regulator of Yorkie. So this increases Yorkie activity, which transcriptionally stimulates downstream transcriptional targets of Yorkie, such as cyclin E and E3 ubiquitin ligase Drosophila inhibitor of apoptosis, which is called the Diap1, to promote cell proliferation and cell apoptosis simultaneously, hence it promotes the tissue growth. So how this Yorkie YAP activation, it propagates throughout this whole tissue, it remains unclear. So we ask this question whether SID, it acts downstream of the Yorkie to regulate wing growth. So to address this, we overexpress Yorkie in the wing disc, which results in overgrown tissues of the sphere or the moderate tissue folding phenotypes. So SID knockdown, it suppressed this sphere tissue folding phenotype, yielding the wing disc relatively much smoother tissues right here. So to this moderate effect of SID RNAi is likely because direct activation of Yorkie is so potent that the resulting ectopic cell proliferation masks the impact of SID RNAi. Thus, Yorkie requires SID function to fully exert its tissue overgrowth phenotype. So in a complementary approach, we decided to activate Yorkie by repressing the upstream hippokinase. In contrast to direct Yorkie overexpression, we expected this approach activates Yorkie at physiologically relevant levels. So we asked whether knocking down SID in this context would also repress Yorkie-mediated tissue overgrowth. So as expected, the hippo knockdown, it resulted in larger wings. So SID RNAi, it suppressed the large wing phenotype of hippo inhibition here. So moreover, we used the heterozygosity of two additional independent SID alleles, which is SID PL00426 or SID deficiency. So it was sufficient to significantly suppress the hippo RNA large wing phenotype. So finally, the Western blot analysis showed that hippo RNAi, it triggers the upregulation of SID protein in the wing tissues. So we conclude that SID regulates wing size downstream of Yorkie. So next, we focused on the mechanism by which SID controls the tissue size downstream of Yorkie. So we examined the expression of hyposignaling transcriptional targets such as cyclin E, Diap1, four-jointed, expanded using standard transcriptional reporter line. So interestingly, expression level of these targets were indistinguishable between wild type and SID RNAi expressing cells. So this is unlikely that SID JIP3 controls wing size by regulating hippotranscriptional program. So, sorry. So then we explored the possibility that SID controls cellular response to hyposignaling via a post-translational mechanism, and we found that it does so. So right here, when we use the Diap1 LEC-Z to determine the expression level of the transcription expression level of Diap1, so it had no effect on Diap1 expression, but immunostaining experiments using Diap1 antibodies, it revealed that there is a significant reduction in the Diap1 protein levels. So additionally, we performed the Western blot assays using lysates prepared from these posteriorly SID knockdown tissues. So compared to the wild type control, SID knockdown in only half of the cell was sufficient to show a reduction of Diap1 protein levels, which confirms our immunostaining experiments. So Diap1 normally promotes cell survival by antagonizing the pro-apoptotic caspase DRONC. So concordant with the loss of Diap1 protein, SID knockdown in posterior compartment cell resulted in ectopic cell death compared to the anterior compartment. So here you can see that more number of cells, they are dying in the posterior compartment compared to the anterior compartment. So these findings, it strongly argue that SID promote wing growth by stabilizing Diap1 activity. So Diap1 protein, it comprises of ring finger domain that has a E3 ubiquitin ligase function. This enables Diap1 to degrade not only downstream substrate, but also itself to modulate cell survival. So ring domain defective Diap1 mutants are homozygous lethal, but heterozygous viable. So we asked this question, to what extent Diap1 ring mutant heterozygosity, it restores Diap1 protein levels in SID knockdown cells. So we took advantage of heterozygous viable Diap1 ring mutation, Diap1-33-1S, and asked whether this heterozygosity rescues Diap1 in SID depleted cells. So you would appreciate that the heterozygosity of Diap1-33-1S allele, it restored Diap1 protein levels in SID RNAi expressing cells. So from here, we conclude that SID, it controls tissue size via Diap1. So this observation that Diap1 ring mutant heterozygosity restored Diap1 protein level in SID knockdown cell, it supports that SID normally stabilizes Diap1 by protecting it from proteolytic degradation. So given the loss of Diap1 protein in SID depleted tissues and the resulting ectopic cell death would increase DRONC activity. So we predicted that SID depleted tissues would sensitive to DRONC dosage such that a partial reduction of DRONC function will be sufficient to restore wing size to some extent. Here you can see that the DRONC, actually it rescues the SID RNAi phenotype. And also the Diap1 ring finger mutant, it also rescues the SID RNAi phenotype, you can see here. So finally, we turn to the mammalian cells to assess whether SID similarly regulates cell survival and cell growth in mammalian tissues. So RNAi knockdown of JIP3 inhibited the growth of HEK293 cell and caused ectopic cell death in flow cytometry assays, mimicking the SID knockdown phenotype in Drosophila. So collectively, our data highlights a novel and conserved SID JIP3 growth control mechanism. Our data, it indicates that SID JIP3 controls tissue size downstream of the Yorkie gap by regulating Diap1 protein turnover. Hence, it modulates cell survival and ultimately tissue size. So I thank you for listening. So, and I'm very thankful to Chabu Lab, my PI, Yves Chabu, my colleague Gangadhar Prasad, who helped me to perform the experiments in mammalian cells, and the graduate students in lab, Nadia Patterson, and the undergrads, Emma, Noah, and Andrew. And I'm very thankful for these gentlemen, Andreas Bergman, Halder, Tian Shu, Herman Steller, and Do Jiap1, for sharing their regions with us. Thank you so much.